Hello, it's Rudain and Nagar, and today we're going to be solving October-November 2021 Paper 1, Variant 1. Let's begin. Question number 1. A student observes a cell using a light microscope. The student then draws the cell. Which items will the student need to calculate the magnification of the drawing? For the student to need to calculate the magnification of the drawing, we need to calculate first the actual size. How to do this is by using definitely an eyepiece graticule as long as it's calibrated with a stage micrometer. Second of all, a ruler. Yes, a ruler is used to measure the image size at this point. 3. Hand lens. Hand lens cannot be used. This is what a hand lens looks like, by the way. It cannot be used to measure magnification. This is incorrect. 4. Stage micrometer. As we said earlier, it is used to calibrate the eyepiece graticule. So this is correct. The answer should be B. Question 2. Which equations correctly show the relationship between magnification, image size, and actual size in microscopy? Let's first draw the equation. Correct answer is going to be 1 and 3, and the answer is A. Question number 3. In which cell structures does DNA transcription occur? Where DNA transcription occurs is where there is DNA. In chloroplast, we have circular DNA. Same with mitochondrion, we have circular DNA. In the nucleus, we have our normal linear DNA. So all three of them can carry out DNA transcription, so the answer is going to be D. Which rows could correctly identify the nucleic acids present in two different virus particles in a sample of air? To start with, DNA and RNA. This is incorrect because a virus either has DNA or RNA. It cannot have both at the same time. And viruses with RNA are called retroviruses. So this is incorrect. 2. It says here that it has DNA but does not have RNA, so could be correct. 3. It doesn't have DNA but has RNA. This is correct. 4. It doesn't have anything at all. This is not possible. So the answer is going to be C. On 4 samples from a mixture of biological molecules gave the results shown in the table. Which biological molecules were in the mixture? To start with, boiled with excess Benedict's solution. Benedict's solution is used to measure the concentration of reducing sugars. So, if it turns from blue to either red or green or yellow, this means that reducing sugar is present in the sample. But in this case, it stayed blue. Therefore, it means that there is no reducing sugar in the sample. So the test is negative for reducing sugars. There are no reducing sugars. So both of these are incorrect. Second of all, boiled with excess Benedict's solution after acid hydrolysis and neutralization. Acid hydrolysis breaks the glycosidic bonds of disaccharides to form reducing sugars. And if the test turns red, after this process, this means that non-reducing sugar was present. So the test is positive for non-reducing sugar. Biuret's reagent is used to test for the presence of proteins, and it turns from blue to purple if the test is positive. Therefore, it means it's positive, and it's positive for proteins, and proteins are present. Here, iodine solution. If iodine test is positive, it will turn from yellow to blue-black color. In this case, it stayed yellow, so starch is not present. The only correct answer could be C. Question number 6. One molecule of X is formed by a single condensation reaction releasing one molecule of water. What is molecule X? If a condensation reaction occurs forming one molecule of water, it means that definitely a disaccharide is going to be formed. The reason why all these rest cannot be correct is because phospholipids form 2 molecules of water. A polysaccharide is a giant molecule, so it could form many molecules of water. Same with triglyceride. A condensation reaction occurs between glycerol and fatty acids, forming 3 molecules of water. So all these cannot be correct, so the only answer is going to be A. 7. The table shows some information about carbohydrate polymers. Which row describes glycogen? As we all know, glycogen is the storage polysaccharide in animals, and it's a highly branched molecule. So let's see the suggestions. First, A is alpha 1 to 4 glycosidic bonds. It does have alpha 1 to 4 glycosidic bonds. B, has alpha 1 to 6 glycosidic bonds. This is also correct because, as we said, it's highly branched, so it has many alpha 1 to 6 glycosidic bonds. Shape of the molecule is branched. Yes, that's correct. Function of the molecule is storage. As we mentioned all these points earlier, so A is definitely the correct answer. 8. The diagrams show 4 phospholipid molecules. Which molecule could contribute to the most fluidity of a cell surface membrane? So the molecule that contributes to the most fluidity in this case is the one that has the most double bonds. The reason for this is that the double bonds prevent the close packing of phospholipids. And if phospholipids don't pack close together, the membrane is normally more fluid. Therefore, here we're looking for the molecule that has the most number of double bonds. In this case, is going to be D. Conversely, the molecule with no double bonds at all forms the least fluid membrane. 9. Which properties of phospholipids explain why single layers of phospholipids are added to water immediately form bilayers? First of all, the phospholipid molecule has a hydrophilic phosphate head and hydrophobic tails. Therefore, they are able to form phospholipids with the hydrophilic part forming hydrogen bonds with water and the hydrophobic center. The hydrophobic fatty acid chains repel water molecules so the tails pack together. This is exactly what we mentioned earlier because fatty acids are hydrophobic and it repels water, contrasting the phospholipid, which forms hydrogen bonds with water. So this is definitely correct. 2. The non-polar fatty acid chains are attracted to each other by hydrophobic interactions. This is correct because they are non-polar and attracted by hydrophobic interactions because they are non-polar. So this is correct. 3. Hydrogen bonds form between the phosphate groups and water. We mentioned this earlier because phosphate is actually polar. So the answer is going to be A. The diagram shows 3 examples of different bonds. Which bonds hold a tertiary structure of proteins together? Here it's specified only tertiary. The only bonds present in tertiary is hydrophobic, disulfide, ionic, and hydrogen. And these bonds are formed by the side chains of the amino acid. 2 is incorrect for this question because it only specifies the bonds in the tertiary structure, and this is a peptide bond. And it is present in all structures, but here it's only specifying tertiary. So this is incorrect. For bond 1, this is correct because this is ionic bond because it's formed by charged amine and carboxylic acid groups. So this is correct. And for bond 3, you could see here sulfur and sulfur. So it forms a disulfide strong covalent bond. This is also correct. The answer is going to be C. Which feature of collagen enables it to fulfill a structural role in skin and in tendons? A. Adjacent collagen molecules are linked by ester bonds. This is incorrect because ester bonds have nothing to do with collagen molecules. They are actually linked together by mostly hydrogen bonds and some covalent bonds. B. Collagen fibers form layers with the fibers in different directions. This is incorrect because they actually form in layers in parallel directions, not in different directions, not a cross-ply structure like cellulose, for example. C. Collagen molecules are formed as triple helix of polypeptide chains. This is correct because 3 polypeptide chains join together by hydrogen and some covalent bonds, forming a single collagen molecule. So this is correct. The graph shows how the concentrations of 4 components, 1, 2, 3, and 4 of an enzyme catalyzed reaction changes with time. Which component is the enzyme substrate complex? So please focus with me on this question. In this line, we could see it's increasing as time goes on. If it's increasing as time goes on, it's definitely going to be the product. And this line, number 3, you could see it decreasing with time. If it's decreasing with time, it's definitely going to be the substrate. So we're going to cross out 1 and 3. Because the substrate never actually reach zero in this case. This means that substrate is in excess. Therefore, the enzyme substrate complex's concentration is going to be constant, and the answer is going to be 4. The reason why we said it's not going to be 2 is because the substrate here is in excess, so the only correct answer is going to be 4 because they are constant. The table shows the Michaelis-Menten constant Km for 3 enzymes. Which interpretation of the information is correct? Enzyme C has a Vmax, which is half of the enzyme P. This is incorrect because here it's only giving you the Km value, which measures the affinity. It has nothing to do with Vmax in this case. So we cannot decide from the data that's given. B. Enzyme C will reach Vmax in the shortest time interval. We can't actually decide that. The enzyme F has the greatest affinity for its substrate. As we see here, the lower the Km value, the higher the affinity. They are inversely proportional. So here, the lowest Km value is going to be F, therefore it's going to have the highest affinity. So C is definitely correct. 14. What explains how a signal molecule produced by one cell can be detected by a target cell? So first of all, let's just quickly explain the process of cell signaling. First of all, a stimulus causes cells to release ligands, and these ligands travels in the blood if it's in case of a hormone. Then point number 3 is ligand binds to cells which have complementary receptors to it, which means that ligands are actually specific to that receptor. And then that ligand activates a G protein, which activates also a second messenger, and then a cascade of reactions is generated in the cell. So let's see the suggestions. A signal molecule can bind to any type of cell surface receptor. This is incorrect because we just mentioned they must be complementary and ligands are always specific. B. A signal molecule has a complementary shape to the cell surface receptor. This is definitely correct. C. Signal molecules enter cell by diffusion. Not always. Actually, if it's only non-polar, it can enter by diffusion, but we cannot guarantee that all signaling molecules are non-polar because there are polar signaling molecules. If the signaling molecule is non-polar, then it could diffuse through the hydrophobic core. So this is incorrect. D. Signal molecules enter the cell by endocytosis. This is also incorrect because it can only enter by endocytosis if it also has a complementary receptor. So the answer is going to be B. 15. Some enzymes are produced in the cells of the pancreas. The enzymes are secreted when required. If they say here the enzymes are secreted when required, this means when a stimulus causes ligands to be released, coming back to the cell signaling process that we discussed in the last question. Which process is used to transport these enzymes out of the cells of the pancreas? Here it says secreted. Secreted means in a vesicle. The only choice we have here is definitely going to be exocytosis, because it also says when required, during only a stimulus. So the answer is going to be D. Plant cells were submerged in a solution with a water potential less negative than that found inside the cells. What describes the conditions of the plant cells after 20 minutes? So it started by saying a less negative. Less negative basically means a higher water potential outside the cell. If there's a higher water potential outside the cell, then water enters the cell by osmosis down water potential gradient. If water enters the cell, it will become turgid. So the answer is going to be D. 17. What will be present in each chromosome at the end of the G2 phase of the cell cycle? At the end of the G2 phase, we know that before that G2 phase, there's something called S phase, where DNA or semi-conservative DNA replication takes place. So the DNA is multiplied by 2. Let's see the suggestions that we have here. 2 centrioles. Centrioles are produced by 2 microtubules lying 90 degrees to each other, and they have nothing to do with the chromosomes. They are found at the 2 poles of the cell during prophase. exactly like this, and they produce the spindle fiber during mitosis. So this is incorrect. B. 2 centromeres. This is incorrect. C. 2 molecules of DNA. As we all know, in each chromosome, each chromatid has 1 molecule of DNA, or 2 polynucleotide strands in each chromatid. So C is definitely going to be the correct answer, because here it's asking me each chromosome, and this is a full chromosome. For D, here says 2 telomeres. 2 telomeres is incorrect, because each chromatid has 2 telomeres. Here it's asking for each chromosome, so each chromosome is going to have 4 telomeres. So this is incorrect. 18. Which events listed are part of the cell cycle? Interphase is definitely part of the cell cycle and is divided into G1, S phase, and G2. And this is correct. Metaphase is at the start of mitosis. This is correct. Cytokinesis happens after mitosis. This is also part of the cell cycle. So the correct answer is going to be A. 19. Which features of an organism are affected by a drug that kills cells that are dividing mitotically? In basic words, in this question they are asking us what are the functions of mitosis. The functions of mitosis we know: repair, or tissue repair, replacement, asexual reproduction, and growth. Let's see the suggestions that we have here. 1. Cell repair. This is the most incorrect answer you could possibly put, because actually a cell does not repair itself. As we said, it's tissue repair, not cell repair. A tissue repairs by stem cells supplying more of that specialized cell, so it replaces the old or dead cells. So this is incorrect. 2. Cell replacement. As I just said, yes, it could replace cells. This is correct. 3. Number of stem cells. Number of stem cells is correct because these are the most rapidly dividing mitotically cells. So this is also correct. 4. Tissue repair. Yes, we just mentioned that. 5. Tumor formation. As we know, cancer cells have a short interphase. Therefore, they divide uncontrollably. So if we kill cells that are dividing mitotically, then definitely cancer cells is going to be the first one. So the answer is going to be D. 20. The graph shows how the distance between sister chromatid, curve P, and the distance between chromatids and centrioles, curve Q, changes during part of mitosis. Which statements could be correct? Number 1. T to V represents interphase. Let's see here. T to V cannot be interphase because, as we see here, the distance between the chromatids increase. The only answer this could be is anaphase. And interphase does not come right before anaphase, so this is incredibly incorrect. The reason why the distances increase in anaphase is because that spindle fiber produced pulls the 2 sister chromatids apart to the poles of the cell, increasing the distance between them. 2. T to W represents metaphase. Metaphase is correct because it comes right before anaphase. We have here PMAT. It comes right before anaphase. W to X represents anaphase. In this case, this is definitely going to be correct, as we mentioned, for the reasons. So as we see here, the answer is going to be B. 21. Which statements help to explain why the 2 sugar phosphate chains in a DNA molecule are at constant distance apart? A. Adenine and thymine are held together by the same number of hydrogen bonds as cytosine and guanine. This is incorrect because adenine and thymine are held by 2 hydrogen bonds and cytosine and guanine are held by 3. So this is totally incorrect. B. Each nucleotide molecule is the same size. This is incorrect because, as we know, we have some nitrogenous bases which are purines These purines have a double ring structure, such as adenine and guanine. And we have pyrimidines, which is a single ring structure. And these are cytosine and thymine. Therefore, nucleotides containing these nitrogenous bases cannot be the same size. So this is incorrect. See, each purine base is linked to a pyrimidine base. This is correct because adenine binds to thymine, cytosine binds with guanine. D, the sugar phosphate strands of the helix are held together by sulfur bridges. This is totally incorrect. As we all know, it's held together by hydrogen bonds, which helps keep that DNA molecule incredibly stable. The only correct answer is going to be C. 22. During DNA replication, what must happen before a newly added nucleotide is bonded to the next nucleotide in the strand? 1. Complementary base pairing. Yes, before the phosphodiester bonds is formed, let's write here, phosphodiester. And this is the bond formed between the two nucleotides. Before they are formed, actually, complementary base pairing must first take place and join together by actual hydrogen bonds first. So they are bonded together by hydrogen bonding. 2. Hydrogen bond formation, as we mentioned earlier, this is also correct. 3. Phosphodiester bond formation. But this is incorrect because it says before a newly added nucleotide is bonded to the next nucleotide in the strand. It is bonded by phosphodiester bond. Therefore, this is incorrect. The correct answer is going to be B. 23. An antibiotic enters bacterial cells through a membrane channel protein P. So, if it says membrane channel proteins, it's either here talking about active transport or facilitated diffusion. Keep that in mind. Some bacterial cells have shown resistance to this antibiotic by acquiring a mutation which alters P, and it's the channel protein. This mutation prevents the entry of the antibiotic into the cell. Which conclusions can be drawn about how resistance to this antibiotic developed in these bacteria? Number 1. The mutation changed the order of amino acids in the gene coding for P. This is incorrect because a mutation does not change the order of amino acids in the gene. It changes the bases in the gene, not the amino acids, so this is incorrect. And by the way, they are changed by either substitution, deletion, or insertion. Let me write them down. Substitution, deletion, insertion. Substitution is swapping a base with another. Deletion is completely removing a base. This causes a frame shift. So all the next bases on forward is actually altered. Same with insertion. It's adding an extra base, causing also a frame shift. And all bases after that mutation happens are altered. 2. The mutation resulted in the production of P with an altered tertiary structure. Yes, this is correct because amino acids have the side chains that determine which bondings takes place. So a mutation could result in a substitution with a different amino acid with the different R groups. And we all know that tertiary structures are held together by bonds. So if the type of bonding is changed, the tertiary structure will also be changed. So this is correct. 3. The antibiotic is a hydrophobic molecule and so cannot pass the phospholipid bilayer to enter the cell. Hydrophobic is incorrect because a hydrophobic actually could pass through the hydrophobic core. So this is incorrect. The answer is going to be D. 24. What is the correct tRNA anticodon coding for the amino acid proline? So, if it's asking you for the tRNA anticodon, and here is giving you the DNA triplet code, there is actually an easier method of doing it. We all know that tRNA don't have thymine. Same with mRNA, they actually have uracil instead. So, it's going to be exactly proline is going to have exactly the same DNA triple code, but what you do, is you swap the thymine with uracil. And here's your answer. No need to convert to mRNA first and do all this fuss. The answer is going to be D. 5. The table shows the observations made by student about 3 different cell types P, Q, and R. Seen on type of cells are P, Q, and R. Let's start. P, it says here structures observed in cell contents, and it says none. If it's none, then it's definitely going to be xylem vessel elements because these are hollow tubes with no organelles. Q, regular pattern of circles in some cells. Regular pattern of circles in some cells indicate that this is the phloem sieve tube elements. Why? Because phloem sieve tube elements have a much lower proportion of organelles. So this is correct. For R, granular appearance of all cells. This is correct because of the sucrose, the amount of sucrose that it has. Because companion cells pump H+ ions and co-transport sucrose inside it, so it could be diffused to the sieve tube elements. Therefore, the answer is going to be D. 20 molecules are attracted to each other. This property is important in the upward movement of water in the xylem. Which term is used to describe the attraction of water molecules to each other? First of all, A, it says adhesion. Adhesion is incorrect because here it says water molecules to each other. Adhesion, water molecules are attracted to cellulose due to its hydrophilic properties and forms hydrogen bonds between them. B, cohesion. Cohesion is definitely correct. Cohesion is the attraction of water molecules to each other because they form hydrogen bonds with each other. The reason for this is water molecules are polar, having a partial negative charge on the oxygen and a partial positive charge on the hydrogens, which allows them to form hydrogen bonds with each other. C, hydrophilic. This is incorrect. This is the reason for the attraction, but it does not describe the attraction. D, polar. Also, this is the reason for the attraction, but it does not describe the attraction. So this is incorrect. The answer is going to be B. Let's start with A. Here it says phloem. And as we know, here it says concentrated sucrose solution. So phloem is definitely going to be W. The reason for this is that the phloem is what transports sucrose and assimilates across the plant. So W is going to be correct for phloem. Let's continue for the xylem. It's definitely going to be X because here it says the water. And xylem transports water and mineral ions. Roots for the moment. Let's move on to leaves. Here we have Z. Z, it says dilute sucrose solution. This cannot be correct. The reason for this is going to be concentrated sucrose solution because in the leaves, this is where photosynthesis takes place, and there is the highest proportion of chloroplasts. Therefore, the most sucrose is produced at the source. So Y must be the leaves. Direction of mass flow along W. As we all know, sucrose moves from a higher sucrose concentration to a lower sucrose concentration down hydrostatic pressure gradient. So it's definitely going to be from Y to Z. So the answer is going to be B. 28. The parts of the heart that control heart action are listed. Sinoatrial node, atrioventricular node, and Purkinje tissue. Sinoatrial node is found in the right atrium and initiates the excitation wave. Next is atrioventricular node, and this delays the impulse 0.1 second. Purkinje tissue, where the impulse is received at the base of the ventricle and the ventricle contracts. Which row is correct for atrial contraction and ventricular contraction? AVN produces waves of excitation. This is incorrect, as we said it's sinoatrial node. B, Purkinje tissue carries waves of excitation. This is correct, it carries it to the base of the ventricle so the ventricle could contract. Now, AVN produces waves of excitation. This is incorrect. C. Sinoatrial node and atrioventricular node produce waves of excitation. This is incorrect. Brings us to D. Sinoatrial node produces waves of excitation. This is correct. And Purkinje tissue carries the waves of excitation. This is also correct. The answer is going to be D. 29. The diagram shows parts of the circulatory system in a mammal. Where is the blood pressure and the speed of flow the lowest? So, we all know that in veins, the blood pressure is the lowest. As we move further from the heart, the blood pressure keeps decreasing. And the lowest speed of flow is going to be the capillaries. The reason for this is the high surface area and very narrow lumen of 7 micrometers. The reason speed of flow in the capillaries specifically is because cells and the capillaries have time to exchange oxygen, carbon dioxide, and nutrients between them. It basically gives them extra time. So the correct answer, as we said for, is going to be the veins with the lowest blood pressure. So the answer is going to be D. And the lowest speed of flow is going to be the capillaries. So the answer is D. The photomicrograph shows human blood cells. What type of blood cell is X? So, let's just start with the basic stuff. If it's a monocyte or a macrophage, it's going to be a bean-shaped nucleus. If it is a neutrophil, it's going to be a granular nucleus. In this case, it's in between. You cannot actually tell the difference. So let's see the suggestions that we have here. A, B lymphocyte. Lymphocyte, we know it's definitely incorrect because in a lymphocyte, it's much smaller than the rest of the white blood cells and the nucleus almost covers the entire cell. So this is incorrect. B, monocyte. As we said, we can't really determine. C, phagocyte. Yes, we can assume it's a phagocyte because phagocyte basically means neutrophils and macrophages, the white blood cells that engulf substances. And D, T lymphocyte. As we said, T lymphocyte cannot be because the nucleus occupies almost the entire cell. So we could just assume it's a phagocyte in this case scenario. The answer is going to be C. 31. The diagram shows some of the reactions of carbon dioxide when it enters the blood from cells in a metabolically active tissue. Which reactions is catalyzed by the enzyme carbonic anhydrase? So the reaction that is catalyzed by carbonic anhydrase happens inside red blood cells, where carbon dioxide plus water are converted to carbonic acid by carbonic anhydrase, and this carbonic acid then dissociates into H+ ions and bicarbonate. Bicarbonate leaves the red blood cell and enters the blood plasma, while the H+ ions stays in the red blood cell. Let's see the suggestions that we have here. As we said, carbonic anhydrase, that reaction happens inside red blood cells, so we know that A and B will be incorrect. We said that it catalyzes the reaction between carbon dioxide and water, forming carbonic acid. So C is going to be correct. The dissociation is not due to carbonic anhydrase, so D is incorrect. The graph shows the oxygen dissociation curves for hemoglobin in animals that live at high altitude and animals that live at low altitude. What explains the oxygen dissociation curve at high altitude? Hemoglobin has a higher affinity for oxygen. This is obvious in the graph because, as you see, in a higher altitude, it combined with oxygen at a lower partial pressure. This means that it has a higher affinity for oxygen. So this is correct. B hemoglobin releases oxygen more readily. This is incorrect because this will only be correct for low altitude because, as you see, it shifted to the right. So this is incorrect. See, the change in partial pressure of carbon dioxide causes a Bohr effect. This is incorrect because here it's talking about high and low altitude. It has nothing to do with Bohr effect in this case. The decrease in percentage of carbon dioxide also to the same point. It has nothing really to do with Bohr's effect, so the only answer is going to be A. 33. The diagram shows three features found in mammalian gas exchange system. Which structures of the gas exchange system should be represented by X in the diagram? So X means it has all three structures: goblet cells, cartilage, ciliated epithelium. The only structures that have all three components is going to be the trachea and bronchus. The only difference is that the trachea has C-shaped cartilage, while bronchus have irregular blocks of cartilage. So the answer is going to be D. 34. What are the short-term effects of nicotine on the cardiovascular system? Actually, nicotine has been removed from the syllabus, but we're going to answer it just in case. Constriction of small arteries. Yes, it does that because it's a stimulant. 2. Increase in heart rate. Yes, it does cause an increase in heart rate. And 3. Increase in blood pressure. This is correct. The answer is going to be A. Some features of a disease are listed. It can be transmitted by animals to other animals, including humans. One mode of transmission is by transfusion with contaminated blood. If it mentioned blood, then we could cross out cholera and tuberculosis because we know they're not transmitted by blood. The only suggestions we have is HIV and malaria. The causative organism can show multiple drug resistance. Let's see the next one. The majority of humans who die from disease are children. As we all know, the majority of humans who die from disease that are children is definitely 100% malaria. It's not HIV. Therefore, the answer is going to be C. Following advice was given to a person traveling to a country where there had been an outbreak of an infectious disease. Cook food well, peel fruit, drink only cool boiled water, wash hands often with soap and boiled water. Which infectious disease would this advice help protect against? Here it says cook food, and here it says peel fruit and vegetables. Drink only cool boiled water. Boiled water, if it's water, then it's transmitted by fecal, oral, root. Therefore, it's going to be because cholera is transmitted by contaminated water. So the answer is going to be A. 37. Which description of multiple drug resistance in bacteria is correct? Bacteria have DNA with resistant genes for several types of antibiotic. This is correct because it says multiple drug resistance in bacteria. So if it has multiple drug resistance, then it definitely has multiple mutations that occurred which gave it the resistant genes for these several antibiotics. Let's see B. Different species of bacteria have DNA with resistant genes for one type of antibiotic. If it's only for one type, then it won't be for multiple drug resistance, so this is incorrect. C. Large numbers of one species of bacteria are immune to several different types of antibiotics. Immune is incorrect. There's no such thing as bacteria is immune. D. Many different species of bacteria are immune. Also, this is incorrect. The only correct answer is going to be A. 38. Which description of a T lymphocyte is correct? They are only found in blood and secrete cytokines in response to infection. Actually, this is not correct because they always present in tissues. B. They can leave the blood and accumulate at sites of inflammation. This is correct. C. They can leave the blood and secrete cytotoxins when exposed to bacteria. They do not secrete cytotoxins, so this is incorrect. Circulate in the blood and always present antigens in response to infections. Lymphocytes are not antigen-presenting cells. Antigen-presenting cells are macrophages or phagocytes in general. So this is incorrect. The answer is going to be B. 39. Where are antibodies found during an immune response? When B lymphocytes mature, the gene inside the nucleus coding for that antibody is used to create B cell receptors. So that antibody gene is used to also create those receptors. So we can say that antibodies are also found on the surface of memory cells, blood plasma. We all know that this is correct because as we know that B cells secrete antibodies in the blood plasma, and these are how they are transmitted in the body. So A is going to be correct. 40. Monoclonal antibodies are used in the diagnosis and treatment of disease. They are produced using a technique known as cell fusion. Which two structures are fused together in this technique? The two structures fused together are myeloma cells and the desired plasma cell, which secretes the desired antibody. The reason for this is plasma cells don't divide, and myeloma cells on their own do not release the antibodies. If they are combined together by a fusogen, substance called a fusogen, they form hybridoma cells, which divide and secrete the required antibody. So let's see the suggestions that we have here. A. Antigens and hybridoma cells. This is incorrect. T. Lymphocytes and cancer cells. This is incorrect. C. Cancer cells and plasma cells. This is correct because myeloma cells are also called cancer cells. Thank you very much for watching this video. If you found this channel useful, please don't forget to subscribe and like and share this video. Thank you.